I'm Julie Wolfrada, Senior Vice President of Development at the Foundation for the National Institutes of Health. I'm pleased to welcome you to the 12th Sayer Vision Research Lecture featuring NEI's Dr. Kapil Bharti, whose work we will hear about in just a few minutes. 15 years ago, Dr. Jane Sayer, a former NIH research scientist at the National Institute of Diabetes and Digestive and Kidney Diseases, established the Sayer Vision Research Lecture and Award with the FNIH in partnership with the National Eye Institute in memory of her parents, Winthrop and Laura Sayer. This lecture series provides a forum for leading scientists with diverse medical specialties to share the latest advances in vision research that could have global significance. I want to thank you, Jane, for your generosity and inspired vision. We at the FNIH are proud of our long-standing collaboration with you and the NEI to promote and advance innovative accomplishments in vision research. Again, thank you all for participating in this event, and please enjoy the 12th Sayer Vision Research Lecture. I'll now turn things over to NEI Director, Dr. Michael Chang.
Well, Julie, thank you very much. I'd just like to add my thanks to Dr. Jane Sayer for establishing this lecture in memory of her parents. And of course, thank you to the Foundation of the NIH for continuing to support this. As Julie mentioned, this Sayer Vision Science Lecture is really meant to feature prominent scientists and give an opportunity to explore areas of interdisciplinary collaboration. And today's speaker, who's Dr. Kapil Bharti, I think is a real superstar, and he's an ideal person to give this lecture. Many of you know that before coming to the NEI, Kapil was originally from India and studied biophysics and biotechnology in college and graduate school there. Then went to Germany to get his Ph.D. in basic biology. Here at the NEI, Kapil has done some beautiful work with a large collaborative team of scientists and clinicians, and really developed the first clinical trial protocol in the US using replacement tissues from patient-derived induced pluripotent stem cells. And specifically, about a year and a half ago, Kapil's team received FDA approval to conduct a clinical trial testing stem cell-derived patch for replacing retinal pigment epithelial cells in patients with geographic atrophy, so-called dry AMD, which is currently not treatable. And as you listen to Kapil's talk, I hope that you'll appreciate, number one, how awesome of an accomplishment this has been, and number two, how it's really required collaboration across a whole range of expertise: stem cell biologists, engineers, surgeons, and really people who develop things like good manufacturing practice protocols. So we're really excited to hear about this. And as a reminder, please submit questions into the Q&A box, and for Kapil afterwards. And don't be afraid to enter them during the talk. So with that, Dr. Kapil Bharti, welcome. We're looking forward to the 12th Sayer Vision Research Lecture.
Thank you, Mike. Thank you for a really kind introduction. And I want to extend my thanks to Julie and the Foundation for their support of this lecture series. But my really sincere thanks to Dr. Jane Sayer for her vision and generosity in starting this lecture series. If I really look at the past awardees for Jane Sayer Lecture, first of all, looking at this list, I really feel humbled. It has Nobel laureates, it has Lasker awardees. Some of my personal heroes are on this. But there are two themes that really jump out to me when I look at this list of scientists, is that pretty much all of them are doing interdisciplinary research and translational research. If you look at one of the last awardees, Dr. Albert Laspada, he's well known for his discovery of trinucleotide repeat extensions and in spinal muscular atrophy. But recently, in collaboration with our own colleagues, Larissa Hearn and Brian Brooks, he has extended his work from neurodegeneration to retinal degeneration, in fact, testing one of the first oligonucleotide therapies for this and showing changes in the vision of these people. If we look at another example, maybe a home example, Ray, one of my friends and colleagues, who has done phenomenal work using 39 ground scroll in synaptogenesis. But I'm most amazed by his recent work, where they really, when they're studying hibernation or cold storage in iPSC-derived neurons from the scroll, and discovered actually the most fundamental cellular respiration phenomena, and from there did a most beautiful translational discovery that I think might revolutionize both organ storage and tissue storage for cell therapy. So these are the kind of people that really inspire us for doing the work that we're doing. Another major inspiration for our work is really the mission of the intramural program. I've listed it here. I'm sure all of you are familiar with this, that really this intramural program fosters and promotes groundbreaking and high throughput, high risk, high reward research, going from basic science all the way clinical to translation using innovative technologies. One of the key things that we have is we respond rapidly to the critical public health needs. And I think we all saw a beautiful example of how NIH participated in COVID-19 vaccine last year. My own lab has started work on hydroxychloroquine toxicity in the eye that I won't be able to talk about today. But really, for me, important is, and you will see that theme throughout my talk, is training of the next generation of scientists that we have here, and trying to promote them to develop their own careers through independent grants that all of them are writing in my lab. Last but not least, really, for us, it's important to do enabling science that enables science through partnership at other academic institutes to transferring technologies to companies. And again, I'll try to give you some examples of all of these approaches that we have taken and incorporated and really embraced the intramural research program for our own research. So for me, the work that we're doing really kind of starts with an observation that almost started 2 decades ago by some of our own, contributed by our own, for instance, Sheldon Miller and Michael Redmond, who really showed that RPE is at the center of this homeostatic unit of photoreceptors and RPE in the choroid at the back of the eye. And this tissue throughout its life, all it does is maintains the health and integrity of the photoreceptors on one side and choroid on the other side. To this audience, I don't really need to tell all the functions that RPE cells do, but I think for me, it's important to emphasize that the way RPE reaches this stage of performing all these things that it does for photoreceptors or for the choroid is through this highly polarized nature of these cells. And it's attained, and that polarized nature by I mean is that it has this very specialized apical structures that talk to the outer segments of photoreceptors, these apical processes. And then it sits on a very unique proteinaceous membrane called Brooks membrane that lets it interact with the choroidal blood supply. It attains this polarized nature by these tight junctions, these proteinaceous complexes that form between neighboring cells, allowing the tissue to become really tight enough that actually in vitro we can measure its transepithelial potential, transepithelial resistance. And these are the kind of, so this, my lab is interested in kind of studying how does the RPE reach this stage? How does the RPE attain these functions? How does it interact with the photoreceptors? How does it interact with the choroid to do all these functions? And then under normal conditions, how does it maintain? But under disease conditions, how do these functions, how are these functions lost? And what can we do for RPE to not lose its ability to support photoreceptors and choroid? And then in cases where actually it has lost, what can we do to regenerate the RPE? So I'll show you some examples of various different activities that we're doing currently in my lab. For us, everything starts with iPS cells. I'm sure all of you are familiar with what iPS cells are, induced pluripotent stem cells. These cells, for all practical purposes, are identical to embryonic stem cells. But the beauty is that we can make them, we can start from any patient's donated fibroblasts or blood cells and reprogram them into iPS cells. This gives us the power of studying human biology in a dish, both developmental biology and disease biology. We take these iPS cells and differentiate them into RPE cells, into choroidal cells and photoreceptors. And that allows us to put them together in a tissue engineering approach so we can try to study all 3 cell types and their interaction in a disease and then connect that with the patient genetics and really come up with kind of both studying the tissue in isolation in 2D or in 3D when all 3 cell types are together, studying what we call disease in a dish when you start with patient cells, so we can follow how the disease starts, initiates and progresses, so that you can intervene at an earlier stage and let the disease and stop disease processes from going further. But in cases where we are too late, we are also working on a regenerative therapy where we try to actually come up with a replacement tissue, the work that Mike just talked about in his introduction. So through my talk, I'll go through, talk about some of the 2D work and then transition to 3D, how we're studying disease in a 3-dimensional complex. And then in the last part of my talk, I'll talk about our cell therapy approaches. So really, for me, all of this started with us trying to optimize differentiation and maturation of RP cells from iPS cells. And this is for me, this is basic developmental biology. We really, the protocol that we developed for making RP cells, mature RP cells from iPS cells, follows basic developmental procedures and principles of how RP cells actually normally form in an embryo. We try to use the right growth factors at the right time point so that, as you can see on top here, cells will go through the normal developmental stages like neuroectoderm, RP progenitors, committed RPE, all the way to mature RP cells in about 10 weeks. And needless to say that in this protocol, we get highly pure cells, as you can see at the bottom left side here, a dish full of pigmented cells. These cells are pigmented, which is what you see here. In the center, you see a flow plot that shows for RP markers that we can obtain more than 98, 99% pure RP cells with a very homogeneous RP phenotype. And at the same time, and you'll see through the application of this process that we have developed, we needed a protocol that is highly reproducible. And I'm glad to tell you that we have, this protocol has worked over more than 500 differentiation runs and over 100 different iPSC lines from different donors, and some examples listed here. And also, again, going back to the enabling stage of our work, that we have shared this protocol with 24 academic labs, with half a dozen companies who have been using it for drug discovery and drug toxicity testing. So I think this protocol is really robust enough that it has survived many hands or has worked in many hands. But for us, we still continue to understand more how the cells are made from iPS cells. And here's an example in the middle. We're trying to now address this in 3 dimensions. We're trying to understand how RP differentiation happens in 3 dimensions. I'll come back to that in a different talk, but there are simple assays we can do to validate the cells. And one of them is just looking at different markers, right? So in this case, what we're doing in green, we're looking at tight junction marker 01, and in red, we're looking at the transcription factor beta-catenin. Beta-catenin is a very interesting transcription factor, which is regulated by canonical wind signaling. Canonical wind signaling is required by RP cells to become RP, but when they become RP, beta-catenin moves out of the nucleus and lines itself around the cell borders, and that's a sign of cell maturity. So we look at beta-catenin and say, hey, is it in the nucleus or is it outside, and tells us whether the cells are mature or they're not mature. Then we can look at more elaborate assays. For instance, in this case, we're looking at electrophysiological response of RP cells, but this assay goes to the heart of the property of these cells, which is the polarized nature of these cells, and it tells us, are they fully mature or are they polarized? For instance, if when the cells come together and they have tight junctions between neighboring cells, they would form resistance to the flow of current. So if we put one electrode on top and one on bottom, we'll see several hundred ohms per square centimeter as a resistance that we can measure here. And as the tight junctions become so tight that they don't allow the receptors and channels on the apical side to exchange with the receptor and channels on the basal side, cells would have differential potential because of different channels on the 2 sides, and that's what we can measure. And then they would have, if they had the right, for instance, potassium channels on the apical side, they would respond to a hyperpolarization pulse, similar to what they do in an eye. And if they would have correct purinergic receptors on the apical side, correct signaling that leads to changes in calcium stores, intracellular calcium stores, they would respond to ATP with this classical biphasic depolarization pulse. So this one assay tells us that so many things are intact and correct with these cells, and they behave very similar to native RP cells, gives us confidence that what we have made in the dish is very similar to patients' eyes or to healthy eyes. But as we try to understand more how these cells are made, and as we kept digging deeper, we realized that how less do we actually know about these cells. And I want to draw your attention to this beautiful paper that was published almost 50 years ago by Roy Steinberg and colleagues, where they looked at what are the different types of RP cells there. By the way, if you're interested in history of science, Roy Steinberg was a postdoctoral mentor of both Sheldon Miller and Paul Seaving, our former directors and scientific director. So in a way, for me, it really come a full circle here from the time where one of the most initial fundamental discoveries in the field of retinal degeneration started, to us looking at that results, those papers again, and trying to replicate them in our model. So what they did here is they used scanning EM and looked at what are the different types of apical processes present on different type of RP cells. And remember, I said apical process is one of the key structure of RP cell that lets it interact with photoreceptor outer segments. And if you look on the left side, it's a hand-drawn drawing of this scanning EM work. What they're showing is that if you know that cone outer segments are smaller than rod outer segments, so RP cells would send out the apical processes tall, and they're like petals of a flower that wrap themselves around the cone outer segment, whereas rod outer segments are short or longer, and they would send that outer segment deep into RP cells. So RP cells have very stubby, small apical processes that are like little fingers that are around the rod outer segments. So obviously you can imagine in a cone-enriched area, RP cells should have one kind of apical process, so-called the petal-like. And in rod-enriched area of the retina, RP cells should have a slightly different kind of apical process, the finger-like. And that's shown here by scanning EM on the right side of the screen. And an interesting thing that have also been noticed by many of the groups is that the macular cells are smaller in size than the peripheral cells. So we asked a very simple question that what kind of RP cells are we making from iPS cells in our dish? You know, are we making one or the other kind? Are we making mixed cells? And to really address that question, so we wanted to first understand what is, if I go back to the size difference, what is our scale bar? What size are we asking for? What size actually in micrometer square is area of macular versus peripheral RPE cells? So to develop a kind of a scale for us, we use an artificial intelligence-based imaging network. And it was a very simple network. What it did was we show it stained RP cells with stained borders, and then it would segment those borders and then assign them based on different size, would actually do shape metrics of all of those cells, and then assign them different parameters or different colors here on this heat map based on are they small, are they medium, or the big in size. And then we can look at the perimeter, we can look how elongated these cells are, we can look at all of these different features of cells. So for instance, here, the darker blue would be the smallest cells, and light blue would be slightly bigger, white even bigger, green and red all the way to bigger cells. So then we use this artificial network, intelligence-based network, and say, what kind of RP cells are present in a human eye? And this is a flat mount of an entire human eye. So this is a single cell resolution complete map, but of 3.5 million RP cells. What you see on this screen is each dot is actually a single cell. It's a very low resolution map that I'm showing you here just because it's so big. But what clearly shows here is that, first of all, we can recognize the macula, dark blue cells, smallest, right? So we replicated these old findings using our network. And then what we immediately saw that this map had a pattern to it. So outside the macula, there were bigger cells. And then there was a ring of slightly smaller cells. This is the optic nerve here, by the way. And then a ring of bigger cells, and then a very peripheral ring of small cells, almost of the same size as the macular cells. So this clearly, first of all, we identified 5 different subpopulations, if you will, of RP cells. And it clearly makes you ask many different questions. So why are the different type of RP cells? Are they differentially sensitive under different disease conditions? Is this map unique to human? Is this map different in different species? So let me try to address some of these questions. This is work in progress. So we're still really quite excited about this and how many different new areas of research this work will open for us. So first of all, this map is unique to each species. We have looked at a few mammalian species now. And the interesting thing is there's a small RP cell, the dark blue RP cell, smaller in area, coincide with high acuity area in most of the species we have seen. For instance, in human here, small cells are in the macula. By the way, this P5 ring that we call, people have described a cone population that more or less coincides with this ring. We don't know the purpose of this yet, but it's interesting that it's similar in size to macula and also coincides with cone photoreceptors. In monkey, we see again the macula we recognize, and then we see a different pattern, first of all, than to human map. And we have kind of a slightly bigger, but kind of a mixed population of RP cell. And if you look at the literature, it shows that the highest density of cones is in the macula in monkeys, and then it's slightly higher density around it, and then it goes down. So it really coincides again with the cone density. We work a lot with the pig model, where we know that there's a visual streak that goes from one end, from temporal to nasal end, and it's enriched in cones. And again, in that area, we see smaller RPE cells. In mouse and rat, There's no high acuity area per se, but we know that cones are distributed, for instance, in mouse more on the temporal and inferior side. And again, we see smaller RP cells on that side. So it's really every time we small RP cells, that is where the cones are going to be. So this again, for me, makes a very, begs a very important and interesting question is this map instructive? So it's a basic fundamental developmental biology question. Is this map instructive for photoreceptor differentiation, or is the photoreceptor differentiation instructing this map? So we're starting to look at this at a most basic level. But going back to kind of the application of this, are these cells different from one another? And are they differentially sensitive in different disease conditions? And so Davide, together with Larissa Hearn in the clinic, put together this table where they actually measured the distance of each of these cell type, starting from the center towards the periphery, so P1 to P5, and then took the ultra wide field images, fundus images of some of the patient eyes, and then did the similar comparison of the actually where the degeneration was seen. And here you see a very interesting example. For instance, in choroidemia, degeneration is primarily in the population P3. In LORD patient, degeneration is in populations P2 and P3. We have, Larissa has a patient with an undiagnosed form of retinal degeneration, where the degeneration is in P3 and P4. Stargardt's we see primarily in the macula, AMD we see in the macula, and interestingly also in P4 and P5. Remember, P5 is where we had identified the smaller ring of RP. So again, for me, this opens many new questions that we can answer. One of the one that we are trying to answer is, what are the molecular and functional differences between different RP populations that dictate differential sensitivity in different disease conditions? And the way, how do we do that? We can't access really these cells from human eyes. So we're good at making RP cells from iPS cells. So we thought we're going to make these different populations from iPS cells. So, and that allows you then to study these different diseases using different populations. I have two projects in the lab that I'm not going to be able to talk about. Just want to mention people who have been working on this. For instance, LORD and Stargardt's. LORD project work is being done by Josh, Ruchi, and Malika, and Stargardt's by Mithra and Devika. So in the interest of time, I'm going to focus totally on AMD today. But you can imagine the application of this process goes both to polygenic diseases like AMD and monogenic diseases like Stargardt's, choroidemia, or LORD. So then, excuse me, how do we make these different cell types from iPS cells? And again, we went back to our artificial intelligence-based algorithm. And what we did here is that we said, okay, so we know how we can identify small and big cells. What if we do a drug screen as the cells were maturing from iPS cells, as the cells were differentiating from iPS cells, and as they become committed RPE, because we can identify their different developmental stages. And the committed RP stage, we treat them with different drugs. And the drugs of choice for us was 110 developmental compounds that up and down regulate all the known developmental pathways, because we wanted to go unbiased, because we didn't know what pathway was going to regulate one or the other cell type. And we wanted to be as comprehensive as possible. So we did a drug screen using an artificial intelligence network. It was immense, what terabytes and terabytes of data that Davide collected to do this work. And after like a lot of going back and forth, optimizing various drug combinations, he actually came up with a cocktail that actually makes in vitro RP cells that have macula specific apical processes versus RP cells that have rod specific apical processes, pure populations. And I hope you can also appreciate in the scanning EM that the macula cells are slightly smaller in size, and the mid peripheral cells are larger in size. In fact, we have the shape data from our AI that we know that they are actually in sizes exactly match those relevant populations. We did the single cell RNA-seq that he analyzed with Ruchi and David to actually show that these cells now have slightly different gene expression patterns. So in blue on top is our P1 population, in orange is our P1 to P2 population, and in green is our P4 population. So we can really, by this treatment, we can segregate them, the gene expression as well. So now, and some of these gene expression actually matches with the published work that people have shown differences between macular and peripheral RP cells. So now we're digging deeper into the physiological differences that there are between these cells. But you can imagine the applications of this that we already are starting to work on, developing disease-specific gene therapy. So I showed you that different diseases affect different cell populations. What if our cell gene therapy was now also tailored to that subpopulation. So this is a work we just started in collaboration with a company called Hubble Therapeutics. And similarly, developing a cell therapy that is disease specific. Again, this is a work that we started with Opsis Therapeutics. I'll show some of our work that we're doing with Opsis in the later part of my talk. But Davide has started asking the question, how are these cells differentially sensitive under different conditions, under different disease conditions? For instance, AMD, and why macular cells are more sensitive to than far peripheral cells, which do not degenerate as quickly. And luckily, he just got a career development grant to do this work from Bright Focus Foundation. And again, I'll show you some examples, other examples of grants that most of my people have been able to secure. And I think that's, to me, it's equally important than doing science, is to really help them become independent in their careers and have them experience of writing grants that we in the intramural program do not write as many. So now what I want to do is transition away to actually show you that once we have all of this collection of cell types, we do also have relevant assays, kind of what I call disease in a dish assays, to study these different cell populations. So I'm going to take an example of AMD here. Many of you know that AMD is a disease, polygenic disease. Many genetic risk factors have been identified. Many of them have been identified here at NEI. There's an environmental contribution contributed by diet, smoking, and aging. And it's thought that all of this together lead to a chronic stress around this photoreceptor RP choroid interface that leads to chronic inflammation, abnormal complement activation, perhaps RP dysfunction leading to drusen formation and atrophy, and in advanced stages leading to dry or wet AMD. So we were interested in focusing on kind of the chronic inflammation part of things. And more so, we were focused on the complement pathway. For those of you not think about complement pathway, there are three different arms of complement pathway: classical, alternate, and lectin. And classical and lectin are known for their role in anti-pathogen. Alternate pathway actually has a broader role, can also be activated through what is called a takeover mechanism. And the way that works is this factor B together with factor D forms the activation of, forms the C3 convertase complex. And that C3 convertase complex breaks it into C3, C3b, and eventually also leads to formation of the downstream convertases, which lead to formation of C5A and C5b. C3b and C5b go towards the MAC complex. So all of this part is common between the three pathways. But C3 and C5a are known to cause inflammatory response in immune cells through the cognate receptors C3AR and C5AR. For us, this pathway, we focus on the alternate pathway because I put here asterisk marks for some of the genes which actually are known to harbor risk alleles for AMD. And you can quickly see that the alternate pathway is enriched for those genes. CF5, which is an inhibitor for the complement pathway, CFH, which is also an inhibitor for this pathway. Both complement the D and B and C3, all of them have risk alleles for this pathway. Immediately you start thinking, well, what is the role of this pathway in AMD? So we thought, how do we address that? And the way we decided to do is to just check if we if we treat our cells with an activated complement versus inactive complement, what happens to RP cells. And here's an example. On the left, you see mature RP cells. This is a slice of cells we stained for affecting that labels the cell borders and the membranes. And ApoE, which is a lipoprotein and well-known marker for drusen, actually, is normally present on the apical surface, apical side of RP cells. But as we treat the cells with active complement, within 48 hours, we see transitioning from apical to basal side, accumulating in the sub-RP space. We put these cells on membrane filters, so all the filter pores are filled with ApoE. So really, and as many of you know, that drusen is one of the hallmarks of AMD. So we can create drusen-like deposits by treating RP cells with active complement. If we start to characterize more, here's on top, H&E image, hematoxylin and eosine stain, we see sub-RP deposits, massive deposits pushing the RP out of the monolayer. And then on the bottom panel, you see cells stained with the dye that looks for oil droplets, and you can see huge accumulation of oil droplets, both intracellular and extracellular in cells that are treated with active complement. So then we're interested in how does that pathway work in RP cells? What's going on here? And as I said, the two anaphylatoxins of this pathway, C3 and C5a, they both act to the cognate receptor C3a R1 and C5a R1. And interestingly, both of these receptors are present on mature RP surface, mostly on the apical side, predominantly on the apical side. So that gave us the idea that, well, perhaps it's working on RP cells through interaction with these receptors. So what we did was to test whether it is the activity of these receptors and this anaphylatoxin that is causing these defects in RP cells. We use what we call serum depletion approach. So what you see on the left side in this graph is we're looking for ApoE deposits. So inactive complement, first bar, to active complement, last bar, you see huge accumulation difference, formation of ApoE deposits. But in between, what you see is complement depleted with serum C3 component or C5 component, or if we block the activity of both of the receptors, we reduce the formation of this complement or ApoE deposits. So suggesting that it is formation of sub-RP deposits is going through the alternate complement pathway. Same thing happens when we look at RP atrophy, measuring transepithelial resistance that I've shown you previously as a feature of RP maturity. Inactive complement to active complement, we see cells atrophying in the presence of complement. But if we deplete our complement of C3, C5, or the serum, we don't cause cells atrophy. So then in the interest of time, I'm just going to summarize here this part of the talk, and I'll show you what I've shown you is, and this is kind of a working hypothesis, is that there's a low-level alternate complement activation that is happening around the RP, also mostly on the apical side in this case, and that has been described by others as what is called the takeover mechanism. And we think that complement factor H and perhaps CFI are able to take care of that low-level complement activation. With age, that complement activation increases because of all those environmental and other genetic risk factors that I talked about. In this case, we mimic that by adding actual complement. So we increase the level of complement that is activated on the apical side of RPE. That leads to sub-threshold levels or over-threshold levels of C3 and C5 that bind to the cognate receptors, activate AKT and ERK kinases that I didn't show you, activate NF-kappa B pathway, downregulate autophagy, and lead to formation of sub-RP deposits at the same time RP atrophy. So that's kind of a working hypothesis that we have. Now, this suggested that if complement factor H was able to take care of low-level complement activation, then even in the absence of overt complement activation in patients, in cells from patients with the CFH risk allele, which as many of you know is one of the highest risk alleles for AMD, we should see some differences in these cells. And in fact, that's what we see. So what you see on this plot is we're looking at lipid deposits comparing RP cells, iPSC-derived RP cells made from patients that do not have the CFH risk alleles versus patients that do have the risk allele. So first of all, even in the inactive complement condition, you see these cells have a slightly higher lipid deposition. And when we activate the complement, that goes even higher in both cases. So this to me suggests that there's low-level complement activation there that CFH was able to take care of, that the risk allele containing version is not able to. And in fact, we do see RP cells secreting C3 and C5, both on the apical and basal sides of cells. So for me, this work really found kind of a proof of concept for doing a broader study that now NEI has started, where we have helped create iPS cell lines with the help of Emily and the ARIS2 patients, making a large panel of iPS cells with various different risk alleles. So what I have listed here is the top 10, 11 lines that we have. If you focus on the middle panel here, the 6 of these, so first of all of these lines are homozygous risk for CFH, CFB, and C3. The 6 of them have homozygous for the HTRA ARMS2 risk allele as well, which is another highest risk allele for AMD. And the other 5 are homozygous non-risk for this category. So it provides a wonderful control comparing these cells. And now we're helping also create syngeneic correction of these risk alleles. And my lab has, and the work that Roba, Devika, Omar, Kelsey, and Ruchi have done have already validated that all of these iPS cell lines do make high quality RPE. So as the people in the community start to use them, they should be confident that these cells will replicate the disease phenotypes they are interested in looking at. But where does this project lead us to? And I don't know if many of you heard the talk that Eric Topol gave last week about the All of Us initiative, which is looking at real life experience and evidence and trying to correlate that with AI to see if there are patterns that would shine from looking at over 1 million people. And he gave many examples of the use of AI across health span. But for me, this project is a very, very small fraction of this kind of initiative where our approach is different. Now we're looking at patient data rather than real life data, comparing it to genomic data and hopefully cellular data coming from iPSC-derived initiative, and using AI to put together all these data sets to see if there are connections that we cannot see in this interdisciplinary approach. So this initiative has already started. Kerry Goods has already started putting together patient data and genomic data into a database, and we hope to contribute cellular data, for instance, the shape metrics data that I talked about. So, and additional genomic data or transcriptomic data coming from iPSC derivatives can be added to this. We are more interested also going into the translational side of things, and going back to that assay for AMD, we did a drug screen and identified, in collaboration with NCATS, identified 3 different drugs that rescued cell death coming from in that AMD assay. And from these 3 drugs, 2 of them were able to suppress both drusen protein deposits and will recover RP health. And these 2 drugs were, one is called L745870, and the other is aminocaproic acid. Aminocaproic acid is a serine protease inhibitor, and we think that is inhibiting the activation or cleavage of complement. But L745870 is a very interesting drug because it is a dopamine receptor antagonist. It blocks the activity of dopamine receptor. Now, if you look at dopamine receptor and also the complement receptors, you immediately notice that both of them are same class of receptors, 7-membrane G protein coupled receptors. And so why this activation of the complement receptor leads to AMD phenotype, whereas a blockage of dopamine receptors inhibits that pathway, tells you that both of them, first of all, have same intracellular signaling happening. So we're trying to identify what scaffolding proteins are regulating the 2 pathways at a common junction point. But then it also begs the question that what is the role of dopamine in AMD, or dopamine receptor in AMD, or the other way around? What is the role of complement receptors in AMD RP physiology? So these are kind of questions that come out of these drug discovery. For me, this is very interesting because we come from a very unbiased approach to a drug that tells us more about RP physiology and AMD phenotypes. At the same time, this drug, we tracked it down. It's a Merck drug that was previously used for schizophrenia. So we're now in conversation with Merck, and hopefully with the help of Emily and Tiernan, we can soon start a clinical trial testing this drug on intermediate AMD patients. But like I said initially, that we're interested in not just 2D disease analysis, we're interested in disease in its 3D context. And here I'm just giving an illustration that in both in dry AMD, advanced AMD stage, and wet AMD, we see that disease affects photoreceptors, RP, and the choroid. Here I want to show you an example of how we're studying RP and choroid interaction to study tissue interaction at the level of advanced stage diseases. So what we want to do is now build a complex RP choroid tissue. And to do that, what we want to do is make use iPS cells to make other cell types. I already gave you an example that we can make RP cells from iPS. But now here, Ruchi with Roba and Tison in my lab managed to make different mesodermal derivatives, which lead to endothelial cells, pericytes, and fibroblasts, some of the 3 components of choroid, also from iPS cells. And then what we do is we try to put this together and try to make blood vessels. So first of all, this work started off as a collaboration with NCATS, and then MJ, Minjay Song, and Russ in my group got a DoD grant to actually do this work. And you'll see that this work is quite expensive. So I'm glad that we get some funding to support this kind of initiative, because optimization of this kind of work took forever. We were trying to make a bioink. So bioink contains hydrogel, endothelial cells, fibroblasts, and pericytes, and we put it together in this bioprinter. These bioprinters have this nozzles or needles that are called the print heads. The beauty of this is they move in a very precise XYZ orientation, and allows us to move to print the bioink in any way we want. Let me try to play this. So in this case, we're printing the bioink in a zigzag pattern, and you can see So this contains all the 3 cell types, and we hope that the cell types would come together and form capillary-like network and then fill the empty spaces by angiogenesis, and we can quantify how much angiogenesis has happened in those empty spaces. So the way this works is we start with a biodegradable scaffold. We flip it on one side, bioprint the bioink on one side of the scaffold, flip it upside down, seed the RP on the other side, and let the scaffold degrade over time as the tissue matures. 2 cell types both make ECM, and the scaffold is now replaced by a Brooks membrane-like structure. And this is how it starts to look like. So this is now, I'm just showing you a choroid. iPSC-derived endothelial cells are labeled in green, pericytes in red. And what you can notice immediately is this capillary-like network throughout the tissue, where pericytes have wrapped themselves around the endothelial cells. You can see they're from the orange color because green and red are together here. Some capillaries are large enough that they go throughout the tissue. And when we see the RP on top, so here's now a tissue that I'll walk from top to bottom. You see RP cells are labeled for tight junction marker, echidna in red, and apical process marker in insulin in green. So first of all, you can see the junction, the monolayer is confluent, and the cells have polarized. As I go down, you will see vessels labeled in PLVAP, which is one of the markers for fenestration. So here's the RP layer, and you can see the confluent vessel network underneath the RP cells. So that's how the 3D tissue looks like. When we start characterizing it further, here on the left side, you see a scanning EM from the side view, RP monolayer, and you can, I hope you can appreciate the Brooks membrane-like structure in between the RP and the capillaries down here. Try to highlight with this pink line. But you can also see the Brooks membrane when we label it for different proteins, fibronectin, laminin, collagen 4, elastin, all of those proteins of Brooks membrane are there, and these cells on top, these are the RP cells. At the bottom, again, side view of TEM by TEM showing RP cells at 3 weeks to 6 weeks. Cells are fully polarized. They have apical processes, tight junctions. Interestingly, they have beautiful basal infoldings that we rarely see when we grow them in a plastic dish. But also you can see the Brooks membrane structure here. And also the tissue is intact functionally. Again, looking at the transepithelial resistance, RP only, or the 3-dimensional 3D tissue has a similar transepithelial resistance. When we look at the choroid side of this tissue, here on the left side, I'm showing you single cell RNA-seq data comparing 2D endothelial cells versus 3D endothelial cells. So first of all, you can appreciate that different gene expression pattern. But when we compared, when we looked at this gene expression more in detail, we realized that in 3D endothelial cells, we had 3 different populations: cells that had higher expression of inflammatory markers, cells that had the higher expression of mature endothelial markers, and the ones that had slightly were differentiated but not as mature. And just to give you an example, that the mature endothelial cells, when we look at in detail, have, for instance, here are some examples listed: KDR, the VEGF receptor, CD34, fenestration marker, and PCAM. And these 2 are also markers for choroid. So these mature cells do become choroidal endothelial cells. And we think that we get these 3 different populations because now if you look at the tissue from the side here, these cells are present at different layers of the choroid. So likely the ones on the top, and we have no way of proving it yet, but likely the ones that are on top are mature choroidal cells that are closer to the RP. But the ones that are buried deep are probably the inflammatory ones because our tissue is not perfused yet. And I think over time, cells undergo senescence as the tissue starts to mature. And hopefully we can actually use that to our own benefit and look at the role of senescence on disease modeling here. But the expression of PLVAP, which is a marker for fenestration, suggested that perhaps these capillaries might also be fenestrated. On top here, I'm showing you again a TEM where pericytes are wrapped themselves around the endothelial vessels. And at the bottom here, you see very fine structures, which are like a small 50 to 100 nanometer pores in the membrane of endothelial cells. These are the fenestrations. So our tissues are indeed fenestrated. And interestingly, the fenestration in these tissues is RP dependent. So what you see on top here is a tissue, intact tissue with RP and capillaries. And with the red is fenestration marker again, and green is endothelial marker. I hope you can appreciate tissues, high angiogenesis and high expression of the fenestration marker here. But at the bottom, in the absence of RP, first of all, the tissue does not proliferate at all. Vessels don't proliferate, and we have a very low expression of the angiogenesis of the fenestration marker. So this is something very similar to what is also seen in dry AMD. Moving forward, Eric has gotten a grant also from Bright Focus Foundation to look at the role of fenestration in AMD and under these different degeneration conditions. So I hope I convinced you that we have structurally a tissue that looks like outer blood-retina barrier with RP and choroid, but we also want to clinically validate our tissue that it does represent a disease condition that is seen in patient. And the disease condition we chose to represent here is CNV, choroidal neovascularization, which is advanced stage of AMD where the blood vessels proliferate and migrate through the RP and leak blood and fluid in the retina. And it's known that that happens because of higher VEGF secretion, and it can be blocked with anti-VEGF antibodies. But it's also suggested that it starts off with hypoxia in the RP, which leads to higher HIF-1 alpha expression, causing higher VEGF secretion. So we asked the question, can we, if we cause hypoxia to the RP, what happens? So as compared to the vehicle on top, if we cause hypoxia to RP cells, we see HIF-1 alpha expression slowly increasing from 48 hours, some cells showing HIF-1 alpha in the nucleus to 2 weeks, pretty much all the cells expressing HIF-1 alpha in the nucleus. And I hope you can also see the monolayer is slowly degenerating. The cells don't maintain their hexagonality anymore. And we also noticed that these cells expressing very high levels of HIF-1 alpha. Now, when we do that experiment in our 3D tissue, so now again, you're looking at it from the side view, RP on top, vessels at the bottom. If we add hypoxia, you see massive proliferation of vessels going towards the RPE and migrating and proliferating in the sub-RPE zone. But we can inhibit that proliferation by adding anti-VEGF antibodies. So really, for me, this is a clinical validation that our tissue does represent a classical advanced AMD and can be used now to further understand disease processes and perhaps discover additional drugs in this context. Moving on, I want to come back to the third part of my talk, going a little slow here, which is to develop our approach to now develop cell therapy in cases where we're not able to stop disease from happening by using drugs, but disease has gone beyond any rescue. Now we have to come back, think of a strategy of regeneration and repair. And for that, our starting approach is to develop RP replacement cell therapy. And this comes from proof of concept that people have done surgically, where they have transplanted an RP patch cut from the corner of the same eye, translocating it into the center, and showing that this transplant in small number of cases where it integrated, it did stop disease from progressing further and actually stabilize photoreceptors from on top of it. And that's the approach we're using to develop an RP patch that we deliver to in what is called the transition zone, where the photoreceptors are still alive, but the RP is gone and stop GA lesion, geographic atrophy lesion from growing further. This is how our RP patch looks like. It's the same scaffold. So this is what I'm showing you is a scanning EM, pseudo-colored, and the scaffold is shown in blue here. You're looking at it from a side angle, you see 3 cells in brown, the nuclei in brown and cytoplasm in brown, but you look at the apical processes of many cells of the monolayer and you see them in green, and you can appreciate a very homogeneous formation of apical processes in this patch. The key challenge for cell therapy is, of course, optimizing GMP manufacturing process, and this is amazing work done by colleagues at CCE in clinical center, and listed people here who have contributed to this. Basically, we had to map out every single step of the process. It takes about 6.5 months, starting from patients, going back to patients, but we have been able to map out from isolation of blood to making iPSC, QC iPSC cells, differentiating them and QC them as they're differentiating, putting them on the scaffold, maturing them and testing them before transplanting in patients. So all of this work, as Mike mentioned, has been approved by the FDA. So we did a lot of testing at this stage to show that our patch is close enough to native RP cells. I'm going to show you just one example in the interest of time, and this is all published work as well. So it's again, going back to artificial intelligence here for a minute. We took advantage of that feature of RP cells, the transepithelial resistance. As I said, that this feature really linearly correlates with maturity of cells. So we fed our neural network images of RP cells as they were maturing and the transepithelial resistance as they were maturing. And once the network was trained, we asked it to predict the transepithelial resistance of the patch just by looking at the images of cells. So what you see on the right side is one of the plots, where on the x-axis is a measured transepithelial resistance, on the y-axis is a predicted transepithelial resistance. We have 3 types of cells: in blue, cells that were not matured at all; in green, with the cells that were partially matured; and in red, with the cells that were fully matured. And I hope you can appreciate that the network can quickly tell the difference, or exactly like measure, like the experiment in the lab, can tell the difference between non-matured, partially matured, and fully matured cells. And both of them have similar results. Once the network was trained, and we're quite excited that the network worked as well, we fed it cells made from AMD patients that it had never seen before. And here's the result at the top, that it correctly predicted the transepithelial resistance of cells from 3 different AMD patients. So we think this is a very easy measure to implement in GMP, and we hope to use it in our clinical setting as well. So moving on, I showed you that we can make RPE patch in GMP. It's functional, but we have to demonstrate that is it safe, and does it integrate into the back of the eye. The safety for this patch was tested in immunocompromised rats. Here we transplanted a 0.5 millimeter RPE patch in the back of a rat, immunocompromised rat. Cells are labeled in red. And first of all, I hope you can appreciate that the scaffold after 10 weeks is completely gone. And interestingly, the entire monolayer patch integrates into the back of the eye, and in fact, to the point that it starts interacting with the rat RPE here. And this to me is a very important point, that the patch, the efficiency of integration of patch is going to be much higher than cell suspension, because it's almost 100% in this case, as opposed to cell suspension where a handful of cells would integrate. So this work led to a massive GLP toxicity and safety study. About 450 rats were transplanted, and we were able to demonstrate that our RP patch is indeed safe and doesn't cause any harm to the animals. So then to move on to testing the patch in its efficacy, we wanted to test the entire 2x4 millimeter patch that we transplant in patients, and to do that, we needed an eye that was big enough of the size of a human eye. So we used pigs in this case, but unfortunately in pigs there's no genetic model that replicates RP degeneration similar to what happens in AMD. So we use micropulse laser and laser ablate the pig RPE cells. And the way this works is the wavelength of this laser is 532 nanometer that is absorbed by the pigment of RPE cells. Cells heat up and burst. As a result, photoreceptors would die off unless we transplant the RPE patch here. So here's an example of the damage done to RPE by laser. On the left side is the infrared image of a pig fundus. There's a 7x4 millimeter defect. Normally the infrared light would not pass through because of the RPE pigment, but in the absence of RPE pigment, you can see kind of a window defect where the RPE cells are gone and the light passes through. By histology, we can show that RPE cells are missing from the Brooks membrane. We see damage to the photoreceptors. The outer segments are thinning here. No damage to the Brooks, to the choroid. The damage to the photoreceptors, I think, is partially because of the laser and partially because of the missing RPE support. So now we want to transplant RPE patch here and have it protect the pig photoreceptors from dying. I'm going to show you a quick video how we transplant this patch in pigs. As you can see, the eye has been prepped, and the surgeon is making incision in the eye wall a little bigger so it can transplant the RPE patch. And in the next shot here, you see the cannula tip. The retina has been lifted and cut, and you can see the RPE patch here, pigmented cells, surgeon is aligning it to the cut in the retina. And in the next shot, you will see the patch that goes underneath the RPE, and then the retina, and the retina is flattened on top of the patch. It's challenging to keep these pigs alive for a long time and immunosuppressed for a long time. We were successful in one pig that we kept for 5 months. Mostly pigs are kept for about 2 months. And we saw beautiful changes happening in the retina. So here, what I'm showing you in OCT, where the red region marks the laser area and white is where the patch is. And you can also see the patch with this bright line. You can see an immune response that happened, probably because of a break in the Brooks membrane. But as you can see that over time, this immune response subsides. This is over 5 months of time we're doing the OCT and imaging. And in that area where the immune response was, there wasn't much changes, or the retina actually degenerated. But look at the retina in the area where there was no immune response and compare it to the laser area. Both the ONL and the autonuclear layer and the internuclear layer we think has been restored in this region just by looking at OCT. So really, the OCT changes are very positive over the area of the patch as compared to where the patch was not delivered. We can also confirm this by multifocal ERG. So if we measure ERG response from healthy regions, we get nice and big response. We measure it on this laser radii where there's no transplant, we get almost no response. And then we measure it from the area where the RP patch was delivered, we get a response that is in between, suggesting that human RP cells are indeed functionally rescuing the pig retina. Once the animals are euthanized, we can also look at them by histology and the integration of cells by histology. Here I'm showing you in green pig photoreceptors and in red human RPE cells. And what we're looking for is whether the human RP cells are phagocytosing the pig outer segments. That would be yellow dots inside human cells. And as you can see, hopefully in this 3D rendering, you can appreciate there's a lot of these yellow dots inside RP cells. That means human RP cells are functionally integrated into the back of the pig eye. Indeed, and they're also rescuing or stopping pig photoreceptors from dying. Here you can see an example of two different areas of the same eye, laser area, no transplant, and laser area with the transplant. Transplant is highlighted with the yellow line. Notice the thickness of autonuclear layer. It's almost double here over the area of the transplant as compared to away from it, suggesting that human RP cells are indeed stopping pig photoreceptors from degenerating. This work was approved by the FDA for a phase 1/2A trial to test autologous iPSC-derived RP patch in AMD patients. I just want to impress upon that this was over 12,000 page IND that I want to thank folks from MS, especially Eric Cardet and Shaker, who are really contributing writing this IND. The clinical trial will have 2 cohorts, first 5 patients with a visual acuity between 2100, 2500, and next 7 between 2080 and 2500. This work, I want to say, was thanks to was accomplished thanks to the grant from NIH Directors Common Fund Challenge Award and the Matching NEI Directors support. Now, the baton for this, as I say, is in the hands of able hands of Henry and Emily, who are very carefully and deliberately selecting the right patients to complete this phase 1/2A trial. Also want to thank Russ and Liz for their support throughout this work, especially Russ, who always keeps reminding me of the mission that we have of doing innovative translational research. And work in my lab is continuing on this track. The 3 very talented women, Francesca, Ruchi, and Mithra, came together and wrote a bench to bedside grant to actually test this approach for another disease indication called choroidemia, and where we are looking at the changes in the choroid using OCTA in these pigs, and also trying to make a choroidemia model where we hope to test the RP patch technology. I'm almost out of time. I want to quickly show you where we are going with this in future. In advanced stages where there is no RP, but in really advanced stages where there's also no photoreceptors, we have to think of more complex therapies. And I've been fortunate to work with the team out of Madison, of David Gamp's lab, and the company that he founded, Lucas Chase's team. And together, recently, we got a huge grant from DoD to reconstruct what we call the entire retina for transplantation purposes. So in this case, what we're doing is we're making both mature, immature RP and photoreceptors from iPS cells and putting them together as a sandwich on one scaffold. It's using the same scaffold that I showed you, PLJ scaffold, RP cells growing on it for a few weeks, and then we put the photoreceptors on top, and then we transplant the sandwich in the same laser model. And again, by looking at these animals by OCT here on the left, control area, no laser, no transplant. On the right is laser area. You can see the damage that the ONL is almost gone and the retina is thinning out. Whereas here you can see the transplant again by a bright line. You can see, you can appreciate that the ONL is rescued in these animals. We have adapted a new technique, adaptive optics, to look at these photoreceptors more closely in these animals longitudinally over time. And hope in this case, we can see that compared to laser eyes with dual transplants, we have a higher number of cone photoreceptors surviving. We can't tell if these are pig photoreceptors or human photoreceptors, but at this point, I think it's preliminary stage. We're excited that we have a recovery one way or the other through the transplant or through regeneration. We do have some changes happening in the retina. And by histology, again, early work, but we can see cells surviving here on the left side. So in all of these panels, human cells are labeled in red. On the left side, RP is labeled in green. You can see here RP cells And then above the RP, you can see some clone opsin labeled cells, again, co-labeled with the human marker, and then a lot of recovering labeling for rods, again, co-labeled with human cells. So we do think that it's at least a good sign that the cells are surviving for almost 2 months in these animals, and so we hope to take it all the way. So with that, excuse me, I want to thank the entire team. I mentioned a lot of people as I went along who were directly involved, but I've been fortunate to work with a much larger team who have many ways contributed to the project. So I want to thank them all. I want to take a moment to actually specifically highlight somebody who has been really, I think, indispensable for this project, and that's Irina Bunia, who is our chief animal tech. I think single-handedly, Irina is responsible for success of all of the preclinical work. So I really want to thank her for boundless dedication to this project. I would be remiss if I did not say some special thanks to some special people that are very close to me. For me, I think the unsung heroes, as I like to call them, of science are the people. We all scientists get acknowledged one way or the other through publications or awards or whatnot, but there are people working behind the doors, behind the scenes, that grease these wheels and make science work smoothly. And for me, those people are equally, if not more important. And the two names that come to top on that list is Kathy Gear and Hiromichia, our two ALMs that have for many, many years done small, large tasks, all of them with a big smile. I want to thank the AMB staff, Travis and his people, our new AO, Renata. As you see, our our work is very data heavy. So ITMB has been heavily involved in many aspects of this. I want to thank ITMB staff. You saw many grants, many cans I had to juggle through. And so Eli at FMB helped me a lot with that. So I want to thank FMB team for that work. And throughout this journey, I've realized that the people who do their job well, who are dedicated to the work, they're important, but they also, I think they're people who are equally and perhaps more important are the ones who have the courage to stick up and say, hey, let's not do this, let's do that, who have the courage to tell me and say, hey, you crazy, this idea is stupid, let's not do this. And at the time when I have a blind spot, they can look at my blind spot. So I've been lucky that I have a couple of such people that I've managed to work with. And I want to thank all three of them, Ruchi Sharma, Arvidas Mamanishkis, and Shekhar Jha. I want to thank Brian Brooks for his support from the day the first day I walked into NEI. I want to thank Santa for her advice and support throughout. I want to thank the OSD staff, David, Mika, Caesar, for the help and support all throughout. Mike, as he recently joined, he has extended his support to this program. And lastly, none of this would have ever been possible if it wasn't for the vision and really relentless mentorship of just one person, and that's Sheldon Miller. So I want to thank him and thank you all. Happy to take any questions. Sorry, I went a little over time.
Well, Kapil, congratulations. And that was really beautiful work and a fantastic talk. In the interest of time, I might suggest that we just take one or two of the questions from the list, and then we can sort of maybe answer the rest offline, but really great job.
Thank you, Mike. Yes, I'm going to do that. Rachel has a question. She's saying, size of RP coinciding with cones is very interesting. Have you looked at all cone retina and does it still hold? Excellent point. That's exactly what we started doing. We're looking at the 13-line ground scroll from Whaley. Unfortunately, because of technical difficulties, we have not been able to make the map yet. We have recently obtained an RL knockout mice from Anand's Root lab, and hopefully we can look at those and answer that question. Great point. Whaley has a question. Could the size of RP cells be correlated with metabolic rate? Any indications from TEGs between populations? I don't have any idea from population studies, but we already have preliminary evidence work that David and Ruchi has been doing that there are metabolic differences between these two populations. As we go on, Rachel has another question, very provocative. Anaphylatoxins as trigger for RP degeneration is, of course, a highly amplified system, likely using consummation of anaphylatoxin that would not be encountered in vivo. Are you planning to make in vivo overexpression model to further examine this question? Again, great point, Rachel. Yes, we just started making transgenic animal models to address this question. So in the interest of time, I'll stop here. Thank you very much, everyone.
Well, Kapil, I think it's actually amazing how much breadth of just science and medicine that you covered here, and just on behalf of everyone at NEI, really, thanks to Julie and Dr. Jane Sayer for sponsoring this lecture, and Kapil, thanks to you for delivering it. Beautiful work. Thank you, and thanks to everyone for attending.